Transportation of oxygen by iron porphyrin. Iron storage. Iron in our body is stored in the liver as ferritin. Ferritin store iron when there is an excess amount in blood or tissues in our body. Iron stored as Fe3+ in hollow sphere shape. It will be released from the ferritin in the form of iron 2+ to other parts of the body. Next, oxygen storage. For your information, there is no long-term storage of oxygen in our body. Basically, oxygen is transported from the lung in the red blood cell, bound to the hemoglobin molecule. From them, it diffuses through the tissues to places where the partial pressure is lower, particularly muscle, where oxygen binds hemoglobin before being consumed. From that, it can be concluded that hemoglobin can be served as short-term storage of oxygen in our body. So, what is hemoglobin? Hemoglobin is a protein found in red blood cell or RBC. Oxygen gas that bind with hemoglobin called oxyhemoglobin, while deoxygenated hemoglobin called deoxyhemoglobin. This hemoglobin functions as a transport mechanism of gases. During inhalation, hemoglobin transports oxygen from the lungs to the tissues of the body. Then, blood gas exchange occurs and body tissues release carbon dioxide back to the lungs. Therefore, exhalation occurs. Hemoglobin is a globular protein and consists of 4 subunits. Each subunit contains a hem group. Each hem group contains an iron atom that is able to coordinate to one oxygen molecule. Hence, each hemoglobin protein can bind 4 oxygen molecules.
Hello. What about iron porphyrin bind with oxygen? What makes deoxyhemoglobin in bent shape becomes planar when oxygenated?
The answer is hem group of hemoglobin is initially in bent shape. Then, come the oxygen with large electron cloud bind to the iron. The oxygen push both of the hem group and become planar. Then, after one oxygen molecule bind with iron porphyrin, it enhances the ability of hemoglobin to bind to more oxygen. And each hemoglobin will attach to 4 oxygen molecules.
What about the buffer effect of hemoglobin when deoxygenated?
For the question. When it becomes deoxygenated, concentration of hydrogen ion is high. And it able to accept additional proton, become positively charged and able to form salt bridges. The salt bridge will be formed between nitrogen and oxygen atom forming deoxyhemoglobin structure. Then, the oxygen can be released. Spectroscopy and the color of blood. The color of oxygenated hemoglobin is bright red and deoxygenated is deep red purple. The shape of the hem group and the hemoglobin protein change, depending on whether hemoglobin is oxygenated or deoxygenated. The oxygenated hemoglobin will absorb light in the blue-green range, and reflect red light. While deoxygenated hemoglobin will absorb light in the orange range, and reflect blue light according to the spectroscopy region.